to Fertile Talks, the global podcast celebrating the diversity of fertility journeys. I'm your host, Dr. Yemi Famuyiwa. Whether you're just starting or simply curious, this is your safe space. In each episode, we will bring you expert insights, heartfelt personal stories, and the latest in reproductive health. Our goal is to break down barriers, tackle stigma, and provide support and hope. So whether you're in the Americas, Africa, or Asia, Fertile Talks is your companion on the path to parenthood. Thank you for tuning in and welcome to Fertile Talks. So the topic for today is there's a lot of genetics when it comes to fertility, and I want to start bringing that up. When we refer to the seed or the soil, the seed being the embryo, the baby, the soil being the uterus or where the seed is implanted. So there are two parts to fertility. Do you have a good seed or do you have a good soil? Now, when people miscarry, it could be because the soil, the uterus, has recognized a bad seed and it's simply spitting it out, right? So it doesn't necessarily mean that the soil has failed. Or conversely, it could be the soil is bad and is not receptive to a good seed, right? So tonight, I'm going to really deal more with the seed, not so much the soil. I think that will be a topic for another presentation because there's a lot to unpack in that. So let's talk about the seed, the embryo, the baby, the birds and the bees. Where do babies come from? So we're going to talk about the genetics of fertility, specifically what makes up the embryo that goes into the soil, right? Most people know, I'm trying to forward this slide. There we go. So most people know that our DNA, what makes us humans versus horses versus crickets, is in the genetic makeup, what's contained in our DNA. That DNA, you usually see a double-stranded swirling mass, is contained in the nucleus of our cells. And we all have sets of this DNA in different, it's packaged differently in what is called the chromosome. The unique thing about human beings is we have 46 sets of chromosomes. We have half from our dad, half from our mother. That's just the way it's written. And this genetic DNA has to split because you can't combine mommy and daddy and combine 46 and get, 92 or something like that. You have to reduce it so that you can now rejoin it to make a new 46. And what we're going to talk about today is how does this come about? What goes into your mom and your dad's gene that results in you? And why are you not the same with your brother or your sister? Why do you have a different eye color, for instance, in a certain family? Of course, your environment does contribute to it, but a lot of it is written in your genetic makeup. So before you make an egg and a sperm, we have to they come from what is known as precursor cells. Those are the so-called progenitor cells that continually multiply to make more of themselves. Now, because this progenitor cells also contain 46 chromosomes, and they divide and still produce 46. And I'll explain how that happens. Right before they make a baby, they have to mature into a fully mature egg or a fully mature sperm. The unique thing with the sperm is the progenitor cells in sperm continues to divide so that you can continue to make more sperm. The progenitor cells for an egg, they stop dividing after a while. So then whatever you have at that point, when the gate closes, that's it. You don't make any more progenitor cells, okay? And those progenitor cells now mature and they unpack to become the fully mature egg. They divide, okay? The process of reducing the DNA content from 46 down to 23 is called meiosis. And when you now reduce it down to 23, you can combine it with sperm that is also 23, fertilize the eggs. This 23 sperm and this 23 DNA from the egg is now going to mix up to produce a new, brand new 46 embryo, which we call the zygote. And that is what now starts to grow rapidly to form the embryo, the baby, okay? So let me reduce this a little bit so I can see better. Shows, okay, I'm just trying to take the viewer thing off my screen so I can control this better, but I can't do it. All right. So how do you break this 46 original progenitor cell down to 23, right? That process is called meiosis. And a unique thing happens when you do that. The first is each pair. So let's say this is chromosome 1, right? And we know we have, 23 chromosomes doubled over to make 46. This original chromosome may have come from mommy. This original one may have come from daddy. This is the progenitor cells. What happens is this chromosome duplicates itself so that it now has two arms. So you see the two arms joined by this ball. It's necessary to produce two arms, and I'll show you why in a second. These two arms exchange genetic material. It's like there's something in the DNA that cuts the DNA, say here, and cuts it here, and moves this over here, and then cuts something over here and moves it over here. Think of it as before you make the egg, God is shuffling the cards. You know when you play poker, you have to shuffle the cards. You don't just deal out the cards. You got to shuffle it up a little bit. Now, because we have 2 of each chromosomes and two sets and 23 chromosomes, if you think about the possible combination that you could shuffle the egg or the sperm into, that's over 8 million possible combinations, right? So no 2 eggs and no 2 sperm can never be alike. It's just not possible because they have to mix it up. Now, when they mix it up, now there's a process that splits and divides this round ball in the middle, divides it up in the middle, and then separates it. So now we are now reduced to separate chromatids, each one bearing only 23 as opposed to 46. So the take-home message is we have to shuffle the DNA, and then we have to reduce the DNA number. Now, if you think of shuffling the deck, what is the possible combination you can get when you fertilize an egg? It's 2 to the power of 46, right? I missed the 46 here. 2 to the power of 46, not 4. And I actually happen to do the math, not by myself, thank you very much. There's something called Google calculation. And you get over 70 trillion possible combinations. Do you understand? That's more than there are human beings on the planet Earth. That's why no 2 siblings can ever be 100% alike, because you're going to get this genetic shuffling. Now, when you get this genetic shuffling, there are obviously things that cut out the chunks, switch them over, and sews it back up. There can be accidents when you do that. So when it doesn't cross over very well. So the accidents some of them can now become inheritable, and some of them can prevent the proper, you get a defective embryo that may not be compatible with life. Okay, so let me explain this in diagram picture. So now remember we had reduced that 23 to little strands. We tore the strands apart, right? So first we duplicated each strand, and then we tore them apart, and then we sent 1 strand from the sperm and 1 strand from the egg, right, to meet up to make 46. There are 46 sets of this now. In order to shuffle the DNA, to shake it up, each strand gets duplicated by itself. So you get this little tongue-like, fork-like thing. And guess what? This is the process I was telling you about, where you sort of exchange little pieces. So you get this piece swings over here, that swings over there, and you can get over 70 trillion combinations when you shake things up. Now remember, When you're shaking things up, there could be accidents, right? So maybe you don't quite shift the entire DNA packet that was supposed to go. So you end up with one extra bit of DNA on one side and one extra bit less on the other side. That can happen. Again, this just explains the process of how the DNA is mixed up, where you cross over, you see that, and sort of exchange DNA, shuffling the deck. This is a summary slide. Don't fuss too much if you don't get this. But what happens, we talked about, this is the sperm, the blue one, having these are the progenitor, the primary germ cells, the original germ cells. They have 46. The cell divides and it has to split this up to make 23 cells each. What it does is it duplicates each length of this DNA here, and then it splits each duplication up, so you end up with 4 arms. In the human egg, most of the genetic material and the cytoplasm, the rest of the cell, ends up with only 1 egg. The rest, it just contains the DNA. We call those polar bodies. They don't really contribute much, right? So it's this one we track down. And this is what combines with a sperm cell to now reestablish your 46 chromosome, right? So a lot of musical chairs going on here, right? So accidents, there are lots of room for accidents to happen. And when they happen, we create what is called aneuploid cells, abnormal embryos that are just genetically so mismatched. And sometimes the abnormality may be so complex that the lab will tell us, oh, wow, it's complex abnormality. We can't even begin to figure it out. Now, sometimes this is a process of the division. When you now create a zygote, this is after it's been fertilized, right? The sperm and the egg has mixed. Now nature takes over. It's now going to break down this one cell into 2 cells, into 4 cells, and then 4 cells into 8 cells within 3 days, and then a rapid phase of division takes place. Now, this part, it's duplicating the DNA, tearing it apart into 2, separating it into 2 cells, surrounding it with cytoplasm, then doing it over and over and over. It's a very rapid process and a rapid rate of growth and division. In the initial part, if you were to separate these 2 cells right here, you could technically form 2 separate individuals, right? Because each cell can make a whole different human being. But you get to the point where the cells now starts to take on specializations. Take home is that the DNA comes from mommy and daddy, and they have to be thoroughly shuffled to create a brand new individual, and there can be mistakes done in the process of shuffling that.
Wow, that was fantastic. Because people wonder, why are some children born with deformities of any sort, or, and stuff like that. And tonight we see why. So is there any way one can prevent these accidents?
In truth, they are very difficult to pretend. You see, when I showed you those strands of DNA that pulled the chromosomes apart to the opposite ends of the nucleus, and then the nucleus divides and you form 2 cells. As a woman gets older, those strands that pull the DNA, they're called the telomeres, those strands. As a woman gets older, those strands tend to break down. That's why when you look at the rate of abnormality, we call that aneuploid. The rate of aneuploidy, the rate of abnormality, let's say in a 30-something-year-old, maybe 20%. But when you get close to 40 and above, that rate of abnormality turns to be 80%, right? So you get more defects, not because the DNA is bad or the egg is bad, but the machinery starts to break apart. It starts to break down. The machinery that pulls those DNA apart becomes defective. So it doesn't do its work as well. That's why the incidence of abnormality rockets up. Thank you for tuning in to Fertile Talks. We hope today's episode has provided you with valuable insights and support for your fertility journey. Remember, you are not alone. Now, if you have any questions or topics you'd like us to cover, please simply leave us a comment And please don't forget to like, share, and subscribe with a friend. Your ratings, your shares, and your reviews are greatly appreciated and is going to help us serve more of our listening audience. Thank you once again. This is Dr. Famiyiwa.